Assalamu alaikum wa rahmatullahi wa barakatuh. Hello, this is Khalid Al-Sayegh. I am going to explain lipoprotein and lipoprotein metabolism in the most, I think, easiest way to explain it. So, lipoprotein is just a way for lipids to move in your body, because lipids is water insoluble structures. We need proteins surrounding these lipids when we eat dietary fat, and the fat goes into our intestines. We need proteins that surround the lipid so it can help and facilitate the lipid transport all the way to the liver, the extrahepatic tissue. We're gonna know all this cycle, and at the end of this lecture, inshallah, you're gonna know exactly what happens to the liver, the lipid, and the route that it gets transferred to. So we said the lipoprotein is a way of the lipids to move in your body because it's water insoluble and the blood is water soluble. So first, I'm gonna talk about the structure of these the structure of these lipoprotein. So the lipoprotein is just a way, as I said, fat that roams around your blood, but it's surrounded by proteins. So it can go and inside your blood to other sites. So let's talk about the structure. The structure, we have proteins, the surrounding part of the lipids, and the lipids. So the lipids, it depends on what type of lipoprotein it is. Is it a triglyceride, triacylglyceride, or is it a cholesterol? It depends, and we're going to see that later on. The proteins are called the apoproteins or the apolipoproteins. It's very important. Very, very important. We're going to talk about it a lot. The apolipoproteins, which is a part of the lipoprotein, there are two major ones. The ones that are structural and transferal. I'm going to show you how. Like, what do I mean by structural apolipoprotein? It is there on one type of lipoprotein, and it's fixed. Or the transferal, it can change. This apoprotein can change from being on a chylomicron to from HDL on being on a chylomicron. It can move from one lipoprotein to another. It means transferal. It can move. But structural, it's fixed. I want to let you know about the lipoproteins, but before we go on and tell you about the lipoproteins and the types of lipoproteins, I want to emphasize the apolipoproteins. The apolipoproteins, the protein component of the lipoprotein, we have basically 5 types, and we have 5 classes, and we have like subclasses. So we have ApoA and ApoB, ApoC, ApoD, ApoE. But the most important ones are in the orange. So the ApoA, this protein, which is a component of a lipoprotein, has ApoA1 and ApoA2. The ApoB, we have ApoB100, ApoB48. We have to know this by heart. It's very important. So the ApoA1 is actually a structural. It's fixed on the HDL. You can't, it's there on the HDL, okay? And the ApoB100 is there on the VLDL, the very low density lipoprotein, which is a type of lipoprotein. I'm going to go through it all over again when I tell you the lipoprotein types, but you have to keep in mind that the lipoprotein have a protein structure component. This protein component have ApoA, ApoB, ApoC. The ApoC, we have ApoC1, ApoC2, 3, and 4. But the most important one, the ApoC, is ApoC2. So what is the function of these lipoproteins? It's there on this is a lipoprotein. But apoprotein, which is surrounding the lipids in the lipoprotein, is important for what? It's actually functions sometimes as a structural component of the lipoprotein, because it will increase the solubility of this lipid when these protein surrounds the lipid. It increases the solubility of this lipid in the blood. So, for example, ApoA1 and ApoB. The apoproteins work as ligands. It's like lock and key. So this is very important function of the apoprotein because we're going to discuss it even later on. To act as a cell surface receptor. So when we have apoprotein on the surface of, let's say, for example, chylomicron, and when this chylomicron reaches a site, this target, let's say, for example, the liver, the chylomicron apoprotein will act as a receptor, will act as a recognition site for this surface receptor on the liver. So this lipid transport protein is very important for recognizing the cell receptors. So apoprotein works as ligands in this situation. So let's say, for example, in the low density lipoprotein receptor on the extrahepatic tissue, not the liver, anywhere else but the liver. Like, for example, the blood vessels. The low density lipoprotein receptors will actually recognize ApoE and ApoB100. But for the liver will actually recognize the ApoA1 on the high density lipoprotein receptor. So after I explain the pathway of the lipid, I'm gonna go over these one more time because I know it's kind of hard to comprehend this information when you don't know exactly what's going on with these lipids. So I just want you to keep in mind that they work as a structural component to increase the solubility, and the other function is ligands for cell surface receptors, so they can recognize the receptor. These apoproteins are very important. And they actually work as a cofactors, as in they activate the enzymes. For example, let's see. I'm gonna go back to this, okay? So I just want you to know that they work as a enzyme activators or cofactors, mean the apoproteins. We're gonna come back to this later on. Now let's talk about the lipoproteins. All of this talking about the apoproteins is actually just an introduction, or I'm just setting on the foundation of this lecture. The apoprotein is a part or a component of the lipoproteins. The other component is the fat, the lipid, whether it's cholesterol or triacylglyceride. Now let's talk about the types of lipoproteins. It's very important to know the types. We have first the chylomicrons, the very low density lipoproteins, the VLDL, the low density lipoproteins, and the high density lipoproteins. Let's say each one and each function, and let's know why are they different from each other. So I want you to focus on this picture. Focus really hard. Now, this is the intestine. When you eat something and the food comes here, then you ate fat, the triglycerides. When they get inside the blood after they go into the lymph and whatever, how does it go transported to the liver? Using a chylomicron. So this type of lipoprotein, the chylomicrons, is the highest in lipids, highest in fat. It's all about triacylglycerides because it came from the dietary food we ate. It's all fat. It's high in TGs. So chylomicron is the first type of lipoproteins. It transfers the dietary fat, triacylglycerides, from the intestine onto the liver. It's dietary fat. You have to know which type of fat, which type of lipids, the dietary lipids. What are they exactly? The triacylglycerides. These chylomicrons is very important. It's the largest in size. أكبر واحدة. The chylomicrons is the largest in size. Why? Is it dense? No, it's the lowest in density. Why do I say it's the lowest in density? because it has very, very, very low proteins, and the triacylglyceride is actually very, very high. So it's the largest in the shape, but lowest in density. So it transferred the dietary fat from the intestine all the way to the liver. I'm gonna tell you how does it get inside the liver using receptors and apoproteins, but now I just want you to focus on each type of lipoprotein and what the main function. So the first type is the chylomicron, and transfers dietary fat from the intestines to the liver. After the chylomicron gets into the liver will take this triacylglyceride and actually synthesize endogenous type of fat. It's also a triacylglyceride, but it's kind of higher than the chylomicrons in proteins. It's called the very low density lipoprotein. So the liver is going to secrete this very low density lipoprotein, the VLDL. So this second type, the VLDL, is first, it's synthesized by the liver. And second, the most important thing, that it is endogenous fat, because it was synthesized by the liver. It's not dietary like the chylomicron, it's different. The VLDL is slightly higher in proteins than the chylomicrons. So it's endogenous TG, endogenous triacylglyceride. So it's kind of smaller in size than the chylomicron, but it also have the TGs, the triacylglyceride, but it's higher in proteins. But it's so it's very low density lipoprotein. If I say density, I'm talking about the proteins. If I say size, I'm talking about the lipid. Keep that in your mind. So in the chylomicrons, the density is like super like low, so it's very low in proteins. Here the density is slightly higher than the chylomicron, higher proteins. As it gets smaller, it gets higher in proteins. So when the VLDL gets out of the liver, containing the endogenous fat, the VLDL will transform into a low density lipoprotein, slightly higher density than the VLDL. It's higher in density, but it doesn't have triacylglycerides like the VLDL. It's derived from the VLDL. The LDL is derived from the VLDL, but it contains cholesterol only. So it contains cholesterol. The low density lipoprotein contains cholesterol. You have to know this by heart. The very low density lipoprotein, the VLDL, contains the triglycerides, but from the liver, and the LDL is derived from the VLDL but contains cholesterol only. So when this cholesterol is derived from the VLDL, and it's called the LDL. So the most important thing about the LDL is it contains cholesterol, and this LDL will transport this cholesterol into the extrahepatic tissue, the peripheral tissue, arteries. That's why we call the LDL the bad cholesterol. So the LDL actually transfer the cholesterol to the extrahepatic tissue, to the peripheral tissue, to the arteries, and deposit this cholesterol, in the one in red, to the arteries. And why? And I mean, it results in atherosclerosis. So when LDL deposits the cholesterol at vessels, it's going to lead to atherosclerosis. So this is the first disease we have today, atherosclerosis. And the LDL, that's why we call it the bad lipoprotein, because it contains cholesterol and deposits onto the peripheral tissue. It doesn't get secreted or anything, it just stays there and results in atherosclerosis. So when this cholesterol is settled down into the vessels, we have the good, the healthy lipoprotein, the HDL, the one in green, as you can see. The HDL, which is the fourth type of lipoprotein, contains cholesterol taken to the liver. So the LDL and HDL are very similar because they both contain cholesterol, but the destination, الوجهة حقتهم وين رايحين, determines whether it was LDL or HDL, determines if it was a good or bad lipoprotein. So the HDL is good, it's healthy lipoprotein because it actually takes the cholesterol from the vessel and transport it back to the liver so it can get excreted through bile and out of the body. So I think now everything is clear. The first one was chylomicron, the first lipoprotein. Only triacylglycerides are transported from the dietary food, the dietary fat, to the liver. And the liver is going to secrete or synthesize the VLDL, which contains the triacylglyceride, but it's an endogenous type of fat. And it's going to give me the LDL, which is derived from the VLDL, but containing only cholesterol. And the LDL is bad because it's going to deposit this cholesterol into the vessels, causing atherosclerosis. And the peripheral tissue cholesterol will be taken up by the HDL back to the liver so it can be secreted into the bile and excreted from the body. Now I want to talk about each type individually, and let's talk about the structure and each function of each lipoprotein. Let's talk and start with the chylomicron.